Good morning. I'm Maria Flynn, and I have a question for you. Do you know someone who has not been able to afford their prescribed medications? You likely do, even if you don't know, because 1 out of every 3 Americans can't afford their medicines. In fact, if they could afford them, they may not be able to afford the most advanced forms. First generic drug approvals bring down the cost of medications. Today, I'm going to be referring to the first generic oral liquids, and those approvals actually done just recently. Oral liquids are very important to vulnerable populations, such as our geriatric populations, our elderly or wise old friends, as well as the youngest, the pediatric populations, and other patients who have swallowability issues. So today, we're going to have oh, there we go. He told me to look panic. So our learning objectives today is we're going to discuss the quality attributes for oral liquids. And we're going to do this in the context and the framework of 2023 approved applications. And so we're going to be discussing 4 case studies. The first 2 case studies, we're going to be discussing the stability of an active pharmaceutical ingredient, also known as an API, or actives. And so the very first case scenario, we're going to be discussing physical stability. And when we think about physical stability, we're actually thinking about the structure of the active. In the second case scenario, we're going to be talking about the chemical reactivity of that active. So if it can degrade, something like that. And in the third case, we're going to be discussing the unexpected leachable situation, as well as our final, which is going to be a container closure risk management scenario. So this slide is showing all the possibles, well, not all, some of the critical quality attributes. You always have to leave room for that. And moving forward, I'm going to be discussing very specifically in the case studies, I'm going to be specifying if it's an oral solution or oral suspension. So when you think about oral liquids, I want to refer you to a chapter in USP. And there's a chapter 1151 that goes through all the different dosage forms for oral liquids. And in that, they'll define each one. So I will not be discussing today the other dosage forms, such as a powder for solution or suspension or syrups, because the last first approvals from 2023 were oral solutions and oral suspensions. So in thinking about the critical quality attributes, we already discussed stability. And why is that so? So when you're in a liquid phase, unlike your tablet where you have your active that's in a fixed position, you have fluidity. You can actually see many things because you're a liquid. So if I were an active, I could see other actives. I can also see other excipients. I could see solvent and all the permeations above. So because of that, they are more reactive. And a lot of times, you will see that you have to have different storage conditions. They might have shorter shelf lives, and they might have some instability. And so we talked about physical stability and chemical stability. And we were looking at assay. We assay the active. And sometimes the active, you can actually see if it is degrading, you can see changes in the assay. But there are also other functional parts of the formulation that we are going to assay as well. Content and uniformity is applicable, but I want to even throw out there another term for oral suspensions, and that's going to be dose uniformity. And so this is very important when you think about suspensions. All throughout the bottle, a lot of the cases, they'll tell you to shake it. And all throughout, you want to make sure from the top, middle, and bottom that you get a homogeneous dose given to the patient. And also over its shelf life, you want to look at dose uniformity. Container closure systems are very important for liquids because they have to be airtight. You don't want to have leakage or spillage. Dosing accuracy is critical in making sure you get the right dose to the right patient for the right indication. Microbial limits. You're in a liquid, so there's water activity that can promote microbial growth. So microbial contamination as well as controls need to be in place. And a lot of times we'll see preservatives being used to be antimicrobial. Leachables are relative to our solid oral dosage forms. They're of higher risk. However, maybe not as much risk as we saw in TNUS when you're dealing with injectables. Dissolution, we're going to reserve to oral suspensions because the active's being suspended, and so you care about it being into how it goes into solution. So let's go ahead and dive into our first case study. When we're thinking about physical stability of oral solutions and suspensions, we're thinking about the solid state. And this is for both dosage forms. You're thinking about the different forms that can occur. The last two, sedimentation and particle size growth, this is only reserved for oral suspensions in that that's what, when you have to shake it, it can either sediment or there could be particle size growth over time. And you need to control that again for dose uniformity. So those will reserve, but I want to also throw out there another comment about redispersibility and resuspendability throughout the shelf life in each dose. So with physical stability of oral solution and liquids, we think about solid state, and we can ask ourselves some questions, for instance, about the active. Is it known to have polymorphism? If it does, there might be an increased risk. We can ask, is the form crystalline? Is it amorphous? Suspensions can have a much greater risk than solutions because in suspension, it's thermodynamically unstable. So inherently, it can have more risk. But whenever you're thinking about different forms, you really need to think about the solution equilibrium of each form. So when we're thinking about solid states of the oral solution, you can think of it lower risk because it's all dissolved if it's a true solution. And if you know that your active is dissolved in a concentration that's less than solution equilibrium, you can think about a lower risk. Now, for oral suspensions, while we mentioned that they're at higher risk, there are some things that you can think of initially that you can think that it may not have as much risk. For instance, if there's no polymorphisms, it's not existing, you don't see it, then, and it's not in the literature, and so far under your control, you don't see it, it may not be a risk. Or you're using the most stable form. And then there's times when all you know is the amorphous form. And in that case, there's a likelihood that there wouldn't be a crystalline form. So that can initially lower your risk. But when we also think about actives, we think about them in terms of solubility. And with solubility, we think about the high solubility class, and we use a class system called BCS, which is the Biopharmaceutical Classification System, and it explains what would be considered highly soluble. So if your active is highly soluble, it's more likely it's not going to have risk. Now, you can increase the risk if you know that you're using the unstable form. And if you also have multiple forms and you know they interconvert, that can also increase your risk. So let's dive into our first case study, and we're dealing with an oral suspension. And this oral suspension is known to have polymorphism. You can see the different forms. It's crystalline, but again, as I mentioned, the active is being suspended, okay? And so there's a greater risk thermodynamically for that. It happens to also be in a risk category of poor solubility, a BCS class 2. So what do they do? What do we do in this situation? Well, you need to mitigate it. Before you even go into formulation and development, all that wonderful things, you need to control your drug substance. You want to make sure you know the polymorphic identification. You want to have a 3-tier particle size for the drug substance. This is before you even get to the drug product. Then, once you know that, you go through manufacturing. Things can happen during manufacturing. In the very end, you get your drug product. So at release, you want to make sure that that polymorphism is what you have. It has not changed. But also, over the shelf life, you want to make sure that it remains in that form. Now, in the drug product also, you want to make sure you have a 3-tier particle size for the actual suspension, that you know what exactly the particle size, and there's a range, and there's 3 tiers. Now, I show you a number that's less than 22 microns, but there are 3 different numbers that make that up. I wanted to put that number there specifically because I wanted to show that this is highly micronized. And this is a trick that pharmaceutical scientists do. We love to, I don't know if I can call myself a pharmaceutical scientist, but I'll put myself in the category since I have 16 years experience. We love to have very highly micronized compounds, because you increase the surface area, which then increases your dissolution rate. For pharmaceutical, so that's you're going into and what you know. But furthermore, since we knew that this fit into such a high risk category, the applicant for this first generic showed a polymorphic study. And they showed that at the 6-month accelerated, as well as at the 24-month, the long term, which happened to be the shelf life for their drug product, there was no conversion. So they had a good control, and they're going to keep all these controls in place and watch it, and so make sure that this does not happen and that they have the form, because it's very important. So now diving into chemical stability, we're going to be talking about different risk. Usually by the time an applicant comes to FDA, they have at least 3 months, usually 6 months of accelerated as well as long-term data. And at that first initial risk, you can either see that there's no trend, or you can see significant trend. If you don't see trend, initially it might have a low risk. That doesn't necessarily mean that will happen over the course of the shelf life, but you can feel a little bit sense of security. When it comes to risk, when you see significant trends, so if you see the assay decreasing, for instance, or you see other degradants forming over time, these significant trends, and especially if they go out of spec, really give you a heads up that there's something unstable about that formulation. It could be obviously we're looking at the active in this particular situation. But there's also tricks that we do in pharmaceutical science that actually stabilize. And so if you see stabilizing agents, for instance, specifically for the API, if you see that going on, then it might give you a heads up that there's a higher risk. So this case study is talking about the API degrading. Even though we're talking about oral suspension here, I can't tell you how many times this come across our desk, where this is universal to all dosage forms. So I put the label here specifically. Sometimes you can get some hints in the RLD labeling about what possibly can go awry, or what can be the failure modes for your formulation or their formulation. So in this particular case, with much evidence I'm not going to show here, the API was thermally unstable, so it was affected by heat. So when you look at the stability conditions, how they knew that this was happening in their specific situation is that they saw at the 6-month accelerated, and I want to mention here, this is a refrigerated suspension. And so the accelerated is at 25 degrees. And they actually saw a significant trend in the assay. And when we say significant, it means greater than 5%. And they also saw out-of-spec degradants. In the long term, the refrigerated, everything was fine. It complied with all the criteria. So the problem is, if you read our guidance the guidance for ICH Q1E, for refrigerated compounds, it's very important that it be supported by real-life data. And so in this case, they wanted 24 months stability or shelf life. They wanted that. And so they needed to back that up with real science. And there's a little bit more going on for the suspension. And so it was very important to see that they had a stable formulation, formula. So we're moving on to our first challenge question. And I want to, sometimes you read these quickly, I want to put your eye in the lens of the API. What is not an active pharmaceutical API-related risk factor for oral liquids? So A, exhibits polymorphism, B, dosing cup inaccuracy, C, poor solubility, D, easily oxidized, air sensitive. So I'm going to give you a couple seconds to think about that. So the answer is B. Dosing cup inaccuracy is a risk. It's just not related to the API. The other three can be failure modes for actives. So moving on, we're going to discuss a case 3 study where you're seeing an out-of-spec degradant. This one's very interesting because only one batch of the 3 exhibit batches failed at 6 months accelerated and at the 24-month long term. In fact, the degradant that they didn't even know they had popped up and started to exceed the limits of ICH-Q3B. I want to refer back to Tina. She had a special case when it came to peptides. They had a much stringent, in this particular situation, they didn't have anything very special about their active, and there was no cohort of concern, but so they were able to use the guidance ICH-Q3B for their drug product. So this degradant it actually exceeded the identification threshold criteria. So under the identification threshold, you don't really need to know, you might want to know, but you don't really need to know what is there because it's considered safe. So what's going on here? They had to figure out what this was because they now need to find out why this one batch had this degradant. And so they identified and characterized, and imagine, this is a first generic approval. So there's no standards. So they needed to confirm this. They do all the wonderful organic chemistry, and they do I'm actually an organic chemist, and so they actually did the structural elucidation, but to really confirm, confirmation of this is the synthetic, actually synthesizing this. So what did they have? Well, they found out it was very similar to an oxidative degradant they already knew about, but it was a diastereomer, which means one of the chiral centers was racemide. And thinking as an organic chemist about how this can happen, it wasn't just like an acid, like the pH of the oral liquid or suspension. Oh, this is actually an oral solution. In that situation, they couldn't really think about how this could really happen. So they had to think a little more. Why did that one batch out of the 3 fail? What happened? What changed? What was different? So they did their homework. What they learned was that one batch, after much thinking, that they were using one normal HCl to adjust at the very end that drug product formulation. And the difference there was the one normal HCl sat in a stainless steel vessel for longer than the other 2 batches. They found that there was iron being leached out of the stainless steel vessel into this acid that is now going to be used for the drug product formulation. And an oxidized reaction catalyzed by the iron can make that degradant. And they went along and did the science and showed that they did have iron that was a higher concentration than the other ones. And they also realistically, theoretically, it made sense. So that explained why this degradant was growing over time and that why they failed stability. So with that in mind, they had a control strategy. They revised their specifications, and they now are controlling this degradant, and they controlled it at the ICH-Q3B. Again, they're allowed to do that. And they committed to even further to do this, since now they understood it, that they would go ahead and they would control this at even a lower limit on commercialization. So what did they do well? They submitted a well-written CAPA, a corrective action preventative action report. They understood the origin of the degradant. They confirmed it by synthesis, and they used a robust analytical methodation to actually see it. And not only that, they committed and they specified the degradant at a tighter limit, not that we had asked them to do that. They chose to do that. And they got first generic approval. So here's our second question, and I know we're short on time, so I'm going to move quickly through this. All are common deficiencies for oral liquids. So what is a common deficiency? Would it be a well-written CAPA report with a control strategy for unexpected finding? Is there an incomplete understanding of the manufacturing process? Is there not specifying the container closure description and integrity? So if you think about this, what is a common deficiency? You know the last 3 are common deficiencies, but a well-written CAPA and understanding an unexpected finding, that's a good thing. So all the other ones would be common deficiencies. So moving right on, now you reversed you're doing a first generic and you reverse the RLD, reverse engineering, you know what's going on, everything's so wonderful. You made this wonderful formulation, and then you choose the wrong container closure system. So we want you always to look at the packaging evaluation and make sure that even over the shelf life, that you are not having any problems with your different configurations. Because in our first case, and our last case, we're going to be discussing this particular picture here, which is 1,000 words. This seal was not appropriate for this single-use unit. And what happened, this is very bad for the patient, they're not going to get the right amount of dose, but furthermore, this could be a hazard for the healthcare professionals and just a mess, depending on what the drug product is. So this was withdrawn. And this first generic approval for the multi-dose containers that had a good seal, they were fine, and they were able to get first generic approval. However, this one is not acceptable. And this is inverted, so when we do these liquid studies, we do the upright, and then we do the worst case. So it could be horizontal or it could be inverted. In this particular case with the single unit dose, it was inverted. So in sum, we discussed the quality attributes and the risk of oral suspensions and liquids. We also discussed risk mitigation possibilities and how they were actually done for 4 case studies in real life for first generic approvals. So I have some closing thoughts. Know your drug product, understand it. Think about the failure modes and the risk. Come up with a mitigation strategy. But when all that fails, expect the unexpected, because the unknown unknowns will occur. But when they do occur, really find the root cause analysis, really do the kappa, and submit that with a very clear control strategy of how you're going to go forward so that you can get a first generic approval. So now I'd like to welcome up Nashwa.